Hi, this is Dr. Kat Flies from Central New Mexico Community College. Video E is the last video on the lymphatic system, and it's a lengthier one compared to the other ones, because now we're going to discuss the secondary lymphoid organs. Remember, in the previous video, we studied the primary lymphoid organs. This included the red bone marrow and the thymus. Primary lymphoid organs represent those structures, organs in which our lymphocytes are born and mature. So that's the red bone marrow and the thymus. All the other organs that belong to the lymphatic system are considered secondary lymphoid organs. Let's take a look at them. Unlike primary lymphoid organs, where lymphocytes originate and mature, such as your red bone marrow and your thymus, secondary lymphoid organs function in accumulating lymphocytes that have not done anything immune-wise yet. But in these secondary lymphoid organs, when they do encounter an antigen for the first time, they will now be able to mount an immune response. So they become immunocompetent, as we say, a word that you should be familiar with. So lymphoid organs are made up of typical structures. For one, we see that they have lymphoid follicles, lymphoid nodules with B and T cells. Typically, they have a stroma made up of reticular fibers, and they have attached to them macrophages. We're going to see germinal centers, except in the spleen. So not in the spleen. We're going to see venules, blood capillary venules, that are very, very, very permeable so that cells from the blood can actually leave easily. And what includes the lymphoid organs, the secondary lymphoid organs that is, are the lymph nodes, the tonsils, the spleen, the Peyer's patches, and MALT. Now, I've already pointed out Peyer's patches and MALT, and since we went over the general features, general anatomy of secondary lymphoid organs, I'm not going to revisit the Peyer's patches and the MALT in this video. Let's get started with the lymph nodes. Lymph nodes are clustered along the lymphatic vessels so that the lymph will be carried from node to node via these lymphatic vessels. And we see them very clustered in or very concentrated in specific regions of the body. So we have a lot of lymph nodes in the cervical region. Remember, when you go see a doctor, he checks your nodes in your neck area. Those are your cervical lymph nodes. And sometimes even when you're really, really sick, you feel that your armpits feel a bit swollen. Those are those axillary lymph nodes that are heavy at work trying to fight off pathogens. You also are going to have lymph nodes in the digestive tract area and definitely in the inguinal area, so in your groin area as well. And there's more, but these are some of the very concentrated areas, and I have those listed right here. Our lymph nodes have two very important functions. For one, they filter our lymph, which is kind of a unique function. In other words, they filter not blood, but lymph. Nobody else does this in our body, or nothing else in our body does this. In addition to that, the cells of our lymph nodes can look for antigens, and when they find antigens, they can trigger an immune response. Lymph nodes are bean-shaped, and they have the typical anatomy of a connective tissue capsule, fibrous capsule that forms trabeculae. So we see these, we see these invaginations here called trabeculae, formed by the fibrous capsule, and these invaginations then form these compartments. The outer layer we call the cortex, which is rich in our lymphocytes as well as dendritic cells. And then the medulla, the inner region of our lymph node, again has B cells and T cells and plasma cells organized in these cords called medullary cords, not illustrated here, kind of like strings of cells, together with macrophages that sit on reticular fibers. So imagine that we have deep within here a bunch of reticular fibers that form the stroma in the medulla. To ensure that the lymph that enters our lymph nodes is filtered well, we will typically see more afferent lymphatic vessels than efferent vessels, right? So this is going to create a bit of a backup within the lymph node, which is a good thing because that means that the lymph hangs out for longer before it leaves. So via or from the afferent lymphatic vessels, we're going to see that the lymph goes next to the sinuses that sit just deep to the capsule, the subcapsular sinuses, which then become smaller and smaller sinuses, and ultimately we see that the lymph leaves via the efferent vessels. So that brings us to the spleen, and that is our biggest lymphoid organ that sits tucked in that hypochondriac region just underneath the ribs. Here you have your diaphragm, here are the ribs. And here we see the kidneys. Sometimes students on cadavers confuse the kidneys with the spleen. So notice that the spleen actually sits more lateral and more superiorly. It is fed by the splenic artery, which branches of the celiac trunk, and it's drained by the splenic vein, which then merges into the hepatic portal vein. Keep that in mind. This is important for you to remember these parts of circulation. Our spleen has many functions. Remember, it's a red blood cell graveyard, and it's also the site where fetal red blood cells are produced because a fetus doesn't have mature red bone marrow yet. Our spleen is also responsible for storing a lot of our platelets. And in addition to that, our lymphocytes proliferate there, lots of immune surveillance and immune attacks, immune responses, and cleansing of our blood. Meaning removal of debris, removal of dead cells, things like that. When we take a look at the spleen, it has a typical hilum, which you learned about, for instance, in the liver, or even the kidneys have it, or your lungs, meaning this is the area where we see the entrance and exit of major blood vessels, nerve vessels, things like that. It's going to be surrounded again by a tough fibrous capsule with invaginations we call trabeculae. And your spleen is going to be made up of lymphocytes, macrophages, but also lots of erythrocytes. Remember, they are taken apart in the spleen. Now, rather than having a distinct cortex and medulla, we talk about the white pulp of the spleen, illustrated here, versus the red pulp. And the red pulp has a nice dark, deep red color in the spleen. The white pulp is mostly made up of lymphocytes that sit on fibers, reticular fibers, of course, while the red pulp is going to be rich in those red blood cells that need to be destroyed, as well as pathogens. That brings us to the tonsils. The tonsils are not the same thing as lymph nodes, by the way. So the tonsils are different structures, and we have a total of 5. So we have in the palate area 2 palatine tonsils that you see illustrated. You see one of them illustrated right here. We also have 2 lingual tonsils near the base of the tongue. So this is the back of the tongue, and this is nearby our palatine tonsils. There we have 2 lingual tonsils, also illustrated right here. And then finally, we have 1 pharyngeal tonsil or pharyngeal tonsil more in the pharynx area, and we see that illustrated on this picture right here. If we open our mouth wide open, we see to either side of the uvula, that little flappy thing dangling off the back of our mouth, there we see the palatine tonsils if they are infected. In addition to these five uh, major tonsils, we also have what we call tubal tonsils that are going to be sitting nearby the opening of our auditory tubes, which are connecting our inner ear with the back of our throat. Sometimes we call them our eustachian tubes. Follicles also have a capsule, like our spleen, like our lymph nodes, but the capsule is interrupted and creates these little crypts, as we call them. So, for instance, right here you see a crypt, and you see more of them here, which allows for all kinds of bacteria and other pathogens to get trapped so that they can be destroyed by the various cells that we find in the follicles with the germinal centers. And you see lots of these follicles with their germinal centers throughout our tonsil. So this wraps up the lymphatic system. Thank you for watching.